Thank you very much. And you may have noticed that in the program, only my maiden name is there. But for the record, I'm still married. I don't know how my husband's name got lost in the registration process. Today I want to give an introduction to different types of gene therapy. And I think, well, you all know Duchenne patients have Duchenne because they don't make dystrophin. And this is, well, a graphic depiction of what dystrophin does. This is a carabiner that you need if you go rock climbing. And the reason I use this to explain what dystrophin does is that you don't need this to go rock climbing. You need this to not fall, well, if you make a mistake. And you don't need dystrophin to have muscle. You don't need dystrophin to use your muscle. You need dystrophin to prevent damage. And Duchenne patients, they don't have a functional dystrophin, and you can see here compared to the carabiner. So if your connection is like this, you're not connected to the rope. Of course, if you fall, there will be problems. Becker patients, they do have these two important domains, so they click to the harness, they click to the rope. Only the elastic thing in the middle is shorter. So if they fall, they will be bruised, but they won't hurt themselves as badly as when this function is not working. So what the dystrophin does, it stabilizes the muscle during contraction by connecting the inside of the muscle fiber to the outside of the muscle fiber. And if you don't have dystrophin, that means you have continuous damage of the muscles. And that leads to chronic inflammation, scar tissue formation, and fibrosis, adipose tissues. Regeneration becomes impaired due to the chronic damage and inflammation. And all these processes lead to gradual loss of muscle tissue and muscle function. And I'm not going to explain what this does to Duchenne patients, because that's, of course, something that you know from firsthand. So since lack of dystrophin underlies all these problems and processes, restoring dystrophin is an obvious therapeutic solution. And there's three approaches that I want to introduce today to restore dystrophin: gene therapy, exon skipping, and genome editing. And I will explain for each how do they work, where are we at the moment, what are the challenges, and also what are the future directions in each. Starting with gene therapy. And gene therapy really is very simple. So Duchenne patients can't make dystrophin because their dystrophin gene doesn't work. So what if we provide an alternative copy that does work? Then patients can make dystrophin from that. However, to deliver genes, you need a vehicle, you need a tool. And most often, what works best to deliver genetic material are viruses. And viruses most often they like the mucus, they like the lung, they like the gastrointestinal, because these are dividing cells. Muscle is usually not a tissue that viruses like. There is an exception, and that's called AAV, adeno-associated virus. That virus is the only virus that can very efficiently go to skeletal muscle. So that is what we need to use if we want to deliver the dystrophin gene. However, it is a virus, and because we have a lot of muscle, we need a lot of virus if we want to treat, because we don't want to reach just one muscle fiber in one muscle. We need to reach all the muscle fibers in all the 700 plus muscles that we have. So you need a lot of viral particles, and that means you can only do this in patients that have not seen AAV before, because if they've seen it before, the immune system will recognize it, and now if you give a gazillion viral particles, there will be a massive immune response, and that is, well, potentially lethal. So that means if you've seen AAV before, you're not eligible for AAV gene therapy. Also, this virus is very small. This is why it can go to muscle, but because it's small, the dystrophin gene, which happens to be the largest gene that we have, doesn't fit. So we need to tinker with it. We need to make it fit, and people have done that. They've created microdystrophins. So we now have a microdystrophin gene to put into the virus that has only the crucial components. It has some viral sequences because the virus or the gene needs to go into the virus particles. It has a promoter, that's a volume switch that says that it's only produced in skeletal muscle, and it has this code for the microdystrophin. Now, what does this microdystrophin look like? We have here the dystrophin protein on top. You can see again the carabiner. So on the left, the actin binding domain, that is that first clicking domain. On the right, you see cis, C-terminus, that is that second clicking domain. And then in the middle, you can see 24 repeats and 4 hinges. Well, that middle domain, that's the elastic part, that is not crucial. You can shorten it. Now, the question is, how much can you shorten it? Well, to make it fit into the AAV, you need to shorten it a lot. And you can see the crucial domains are there, the clicking domains, but this elastic domain in the middle now is really short. It's shorter than it is even in Becker patients. And it has either 2 or 3 of these hinges and 4 or 5 of these repeat domains. In animal models, this is functional, and if you treat animal models that don't make dystrophin, this will improve their muscle function and muscle breakdown. So it's functional, but it's not fully functional. So these viruses, these microdystrophin viruses have been developed, and there's currently 4 that are in clinical trial by Solid, Pfizer, Sarepta, and Roche. They collaborate, and Genethon. They use 3 different types of AAV, 7.4, 8, and 9. And combined, roughly 100 patients now have been treated. And from this, we know some general findings. So we know that if you do systemic treatment, so into the bloodstream, that microdystrophin will be expressed in the skeletal muscles. The levels differ. So it's anywhere between 5% of all the fibers to 80% of all the fibers. And if you look at the absolute numbers, about 1 to 74% of normal dystrophin. However, there's also been side effects, and these have been severe to very severe, and sadly 1 patient so far has died after receiving AAV gene therapy. The severity of the side effect is related to the dose. However, we cannot really decrease the dose because this 5% and this 1%, those are achieved with the lower doses, while the 80% and the 74%, those are achieved with the higher doses. So we're at this, say, very critical level where if we lower the dose, then it will be less efficient, but we can't increase the dose because then it becomes more toxic, and at the dose where we're at already there is a risk of side effects. It's also age-related, and probably older people have a more mature immune response, and that gives the side effect. But also if you're older, you generally will be heavier, and you get the viruses per kilogram body weight. So if you're older and heavier, you get more viruses, and that probably also will lead to more severe side effects. There's 2 types of side effects. So the ones that I just mentioned are the acute ones. So they arrive shortly after being treated with the AAVs. They involve severe nausea, they can involve liver damage, kidney failure, sepsis, or death. And this doesn't happen in all the patients, so I don't want to paint that picture. But it has happened in patients, so I also don't want to gloss over the risks, because these are, of course, severe risks. There's also another type of side effect that occurs later in a few patients, and that is muscle breakdown, so rhabdomyolysis in the technical term, and myocarditis, so an inflammation of the heart. And this is due to an immune response to the microdystrophin. And it's only seen in patients who have the lesions at the start of the gene. Now, how does this happen? Because Duchenne patients don't make dystrophin. So dystrophin for them is new. And proteins that are new normally are recognized by our immune system. That's how they recognize bacteria and viruses. So why don't all patients have an immune response to the microdystrophin? Why only this subgroup? Well, there's no clear answer yet, but this is my understanding of it. So we have at the bottom the whole dystrophin, and then at the top we have that small dystrophin. And you can see the small dystrophin, it has this first click domain, it has the last click domain, and a very small elastic domain. That is based on the dystrophin that we have in muscle, the one at the bottom. However, all of us also make a very short dystrophin that only contains the last click domain. And we don't make it in muscle, we make it in all our other cells. Now, this protein is also made by 99% of the Duchenne patients, because it's at the very end of the gene, and most of the mutations are before. So this protein can be made by most Duchenne patients. So they know this protein. They know that part of the protein they know because they're making it. So that will not trigger an immune response. Now, at the beginning, so most patients will have a mutation somewhere there. in the middle of this elastic domain. That means that the beginning of the protein is produced. It's not stable, it's not functional, but the immune system will have seen it. So also for most patients, that part of the protein is not new. They have seen it, it's just normally not functional. Now patients who have a deletion at the beginning of the gene, they will not have made that first part because it's deleted. And now when you come in with the microdystrophin that suddenly has that part, for them that is new. And then an immune response can happen, and then you can start breaking down the muscles and the heart that express that. So that is well, not something that we thought would happen, but that does mean now that sadly patients who have these large deletions at the beginning of the gene are excluded from gene therapy because the risk of them developing this is too severe. So this is, well, bad news, obviously, but there's also some other things that we don't really know. So how functional are these microdystrophins? We know they're functional in mice and dogs, but are they also functional in humans? And well, what that mouse does is not how mouse normally walk. They walk on 4 legs. We walk on 2 legs. So the dynamics will be different. And then the question is the microdystrophin as functional in humans as it is in mice and dogs? And there's currently placebo-controlled clinical trials ongoing, and then we will really know whether it's functional or not. What we do know is it won't stay there forever because it's not fully functional. So with time there will still be muscle turnover. And with muscle turnover, you will lose the microdystrophin because AAV doesn't go into the nucleus, it doesn't go into the genome, it will just float around. So with time you will lose it. And then the question is, will that happen in 2 years? That would be a problem. Or will it happen in 20 or 30 years, which would be much less of a problem? What we do know is, because of an immune response to the virus that you will get from the treatment, retreating will be very difficult because obviously you will have an immune response after you've treated patients once. And then there's this last issue of the immune response to the microdystrophin. This is something that people are studying because it's not seen in all patients with deletions in the beginning of the gene, only in some. So why do some patients have this? Why do others not have it? And if we can understand that, then we can also better select, because you don't want to treat the patients that will get this, but you also don't want to not treat the patients that would not get it. So there's research to be done, and I think this is the thing, that if you're pioneering something, so this is the first time that at this number people are being treated with this microdystrophin gene therapy, new questions will arise, and sadly that's generally how it works in science. So what are people doing to make this better? They're optimizing, so they're trying to optimize AAV so that it goes better to muscle. Because if you can do that, you can lower the dose. And then if you can lower the dose, the side effects will be much less. So that would be safer. People are also working on other ways. So how can we make it, how can we do it without a virus, so non-viral delivery? Can we use nanoparticles? Well, sadly, nanoparticles, nano means very small, but they're still too big to go to muscle. So we need femtoparticles, if you like. And that's also something people are looking into, but that's at an earlier stage. And then if you want to repeat delivery, well, there's different types of AAV, but the question is, they're still all AAV. So how much flexibility is there? If you've once been treated with AAV9 and you need retreatment, can you then be treated with 7.4, for instance? So that is something that's being studied as well. And then if we make the nanoparticles work, that would allow retreatment as well. So then we have exon skipping, that's the next one. And what exon skipping tries to do is it tries to allow Duchenne patients to make a Becker-like dystrophin. So what we have here is a deletion of exon 48 to 50. And that's a very common deletion in Duchenne patients. And you see 47 doesn't fit to 51. And that is because Duchenne patients have mutations that disrupt the code. So it doesn't fit, the code becomes unreadable, and the translation from genetic code to protein stops. You can also see 47 would fit to 52. And that's actually a deletion of exon 48 to 51. That's a Becker-type deletion. And how does that work? If you have 47 to 52, the code remains readable. You'll miss something in the middle, but we've already seen as long as you have these 2 clicker domains, the protein will be functional. So when the code needs to be translated First, these exon things are linked together in a process called splicing. And what we here want to do, we want to manipulate that process. So we want 47, instead of being connected to 51, which would normally happen, we want it to be connected to 52. And how do we do that? We do that by hiding exon 51 with an AON, an antisense oligonucleotide. And that is a small piece of modified RNA that specifically recognizes exon 51, hides it, and now instead of linking 47 to 51, 47 is linked to 52, the code becomes readable, and a Becker-like dystrophin can be produced rather than a non-functional dystrophin. Now this is a mutation-specific approach, so depending on where your mutation is, you need the skipping of a specific exon. And also for some mutation, exon skipping sadly will not work, because, for instance, the mutations are in one of these crucial domains. Now, exon 51 skipping would apply to 14%. If you then also skip exon 45, you skip an additional 9%, and so on and so forth. But you can see these are subsets of patients. So where are we at now? There's 4 AONs approved in the US. Not in Europe, not in the UK, they're only approved in the US. They target exon 51, 53, and 45, and you say that's 3 exons? Yes, because there's 2 AONs for exon 53. And that's about 30% of the patients. This is only based on dystrophin restoration at very low level, generally around 1%. So what the FDA has asked the companies that develop these AONs to do is to collect data to confirm that these very low dystrophin levels actually slow down disease progressions. And that's important because, well, first of all, the drug is very expensive, but also these AONs don't have eternal life, and they target something that also doesn't persist forever, so you need repeated treatment. You need weekly intravenous dosing. So what is clear is that there's room for improvement. Less frequent dosing would be nice, but also higher dystrophin levels would be nice. And then there's 2 types, and in view of time, I will not go into applicability. I'll also only go into improving the delivery to the skeletal muscle and the heart, because why are these levels so low? Because we don't get enough of these AONs into skeletal muscle and heart. And there's different ways to improve this. You can add a conjugate, so-called arginine-rich conjugates, and these improve delivery to all tissues. So not muscle-specific, but it improves delivery to all tissues, so also to skeletal muscle and heart. And there's 3 companies that work on this. Sarepta, they have the SRP-5051. That's eteplirsen, that's their 51 oligonucleotide or AON that is approved, now linked to one of these peptides. PepGen has one targeting exon 51, and Entrada, they have a cyclic peptide. And why do they have a cyclic peptide? The others have a linear peptide, because they claim that is safer. But they've not yet tested it in clinical trials, so we don't know yet whether that really is the case. So are there safety concerns? Yes, there are, because what you see in the Sarepta and the PepGen, Sarepta has tested in patients, PepGen in healthy volunteers, and they see hypomagnesemia. What does that mean? There's too little magnesium in the blood, probably because there's too much magnesium excreted through the kidney in the urine. Patients are being supplemented with magnesium so that the serum levels don't come too low. But the question is, of course, why do the kidneys excrete this rather than reabsorb it? And is this a sign of kidney toxicity or not? And if it is kidney toxicity, can we do this long term, yes or no? So these are things that we don't know yet. And we also don't know yet whether the Entrada one will actually be safer or not. So outstanding questions again. So there's also companies working on trying to improve to skeletal muscle only, so not to all tissues. And they do that by using an antibody against transferrin, and transferrin is expressed on skeletal muscle. So it's like, well, writing an address, go to muscle. There's 2 companies working on this, Dyne and Avidity, targeting exon 51 and 44. And then, well, Dyne has just started their clinical trial. Avidity is still preparing for trials. And there's, well, we don't know yet whether this will work. In animal models, it seems to work, but, well, everything seems to work in animal models sometimes. are there antibodies against the antibodies? Is that possible? These peptides from the previous slides are really short, so probably too short to get an immune response. Here, this antibody is larger, so will there be antibodies against the antibody? And if that happens, will that be a problem or not? Transferrin is expressed highly on muscle, but it's also expressed on liver and brain. Will that be an issue or not? I mean, there's also brain problems in Duchenne, so maybe it's not a problem, but we don't know yet. And the final problem is manufacturing is much more challenging because this antibody is quite big, so it's much more challenging. And will that be an issue? Or because now more and more people want it, the manufacturing is optimized and it stops being a problem. And then one thing that you can think of, well, what if we combine gene therapy with exon skipping? Well, there's a way to do that. You can deliver a so-called antisense gene, and then you would have to treat only once. You wouldn't have to treat every week. And Kevin Flanagan is doing this, and the antisense gene is called U7SNRNA. And he's done this for 3 patients with exon 2 duplications. And in 2 patients, he's seen dystrophin restorations, I think, around 5–8%, so not that much if you compare it to the microdystrophin. But for one patient, a 7-month-old patient, he's seen close to 100% of dystrophin after doing this. But only 3 patients, so yeah, very limited numbers. And there's currently no company that really works on this. Audentes was working on it. They were then acquired by Astellas, and at some point Astellas said, well, we need to prioritize, and they stopped this preclinical development. So there is a gap, and hopefully other companies will pick up on this and start working on it. But of course here, all the challenges that we had with gene therapy for the safety, etc., they also apply here because you're now using AAV to deliver the antisense gene rather than the very small antisense oligonucleotides that are easier to work with. I'll skip this in view of time, and I'll move finally to genome editing. You've probably all heard of it. My husband is an engineer, nothing to do with molecular biology. He knows what genome editing and CRISPR-Cas is because it's in the news. Well, what is it? Very simply put, it's a scissor that cuts the DNA and a GPS that tells the scissor where to cut the DNA. Now, why on earth would you want to cut the DNA? You want to do this to activate the DNA repair system. If you have dividing cells, you can do amazing things. However, skeletal muscles are not dividing cells. So we're stuck with what's on the right. The technical term is non-homologous end joining. I call it the glue system because what happens is the 2 pieces are just put together and glued. And this causes mutations because, well, part of you get the break and the DNA starts degrading. Some DNA building blocks might be floating around there that the glue system thinks, well, you're here, you're probably used to be here, I'll put you in. So this causes mistakes. Why would you want to cause mistakes? Because with mistakes, you can do permanent exon skipping. So rather than, again, these weekly intravenous infusions, you might have to treat just once. So how can you do this? You can use 2 scissors. And we have here a deletion of exon 52. You see 51 doesn't fit to 53, but 51 fits to 54. If you have 2 scissors, you cut on each side of exon 51. 51 will be gone, the 2 ends will be put together, and then you end up with a code that is now readable, and every time you make a transcript of this code, it is repaired. And the AONs, they target this process, and therefore you need to do it every week. Another way of doing it is with 1 scissor. Then you cut the exon, and because the repair is not it induces errors, the exon becomes invisible. So it's there, but when the exons are linked together, it's not seen, and again you get a repair. What's the advantage of this is you need only 1 scissor. The more scissors you need, the more chance there is that weird things start happening. So genome editing is hyped, and like I said, everyone has heard of it because the news is all over it. It got the Nobel Prize. It got the Nobel Prize for chemistry, not for medicine. I think that's very important to remember. And why are people so enthusiastic about it? Because for the first time, us scientists can do things that we couldn't do before. We can make cell and animal models that used to be impossible, or we can make them in less than a year, where it used to take 10 years. So that all is very nice and helps also therapeutic development. And then, of course, when that happens, people start thinking, well, if you can do it that easily, can't we also use it as a therapy? And of course, in theory, we can, and there's publications out there that show that it's possible in cultured cells from Duchenne patients in animal models that don't have dystrophin. There's even clinical trials ongoing for patients with liver diseases. So isn't it possible to also do this for Duchenne? But there are some things that need to be solved. So why are people doing this for a liver disease? Because you can deliver with lipid nanoparticles to the liver very safely, and you can then deliver transient scissors, not permanent scissors. If you use AAV, you deliver permanent scissors. So they will do their job, they will cut where they're supposed to cut, and then what will they do? They stick around forever. Will they randomly cut somewhere else? Well, that's not what you want. And also, it's a two-component system. This scissor gene is very big, so it needs its own AAV, and then you need your other AAV for your guide. And that means you get a dilution of your effect. And normally, I said, normally AAV doesn't go into the genome. However, if you start cutting the genome and there's AAV genes floating around, it will end up in the genome, and that's another safety concern. What's also important is, so some people like to say CRISPR cure. Sadly, for Duchenne, that is not what will happen, because what you get is a partially functional dystrophin. You don't get a fully functional dystrophin. So when people say correct the mutation, what they mean is reframe, allowing the production of a partially functional protein. And, very important, the time of intervention will matter. When people say cure, I think Jesus curing people, people that didn't walk started to walk. That will not work here. If you allow a muscle to produce dystrophin and all the muscle is replaced, that will not restore the muscle that is lost. And I wish I could tell you something differently, but that's sadly just not what's currently feasible. So the time of intervention will matter. Function that's lost will not come back. Now, the safety I mentioned already. How specific are these scissors? What will happen if they cut somewhere else? This is the DNA. There's no undo button, so that is something that needs to be solved. And again, in view of time, I will skip over this as well. And I'll go to the last point before my summary slide. So, dystrophin restoration is not enough. I mentioned the accumulated damage. With time, the muscle is damaged. So you need to also target the muscle damage. So what most likely will happen is a combination therapy. One to restore the dystrophin, but then also another one to improve muscle function, or maybe multiple ones. And I'm mentioning here the Givinostat, not because I have stock or anything in that company, because that the Ital Pharmaco clinical trial had positive phase 3 results, so that's why I'm mentioning it here. But there's lots of other compounds in development that also try to improve muscle quality, and that hopefully in future can be combined with dystrophin restoring therapies. So, in summary, it's possible to restore dystrophin with exon skipping and microdystrophin. We've seen that in patients. It's also clear that there's room for improvement. The genome editing is still in the preclinical state, and most likely you need a combination therapy. And I've focused here on the challenges, and well, I don't want you to lose all the hope, because I think the thing is we're pioneering lots of things. We're discovering things that we didn't realize while we're working on it. But the good news is that there's a lot of very dedicated scientists working on all these things in parallel and also collaborating. So there's still a lot of questions, but we also are dedicated to try and make these things working for you and your sons. So that is my last slide. I'm not sure whether we have 2 talks and then questions, or whether we have questions now. I wasn't here yesterday, so I don't know the setup. Linda says 2 talks. Now, I'm.
We'll just keep it to 5 minutes.
5 minute questions, and then we switch to Linda. And I wasn't here sadly yesterday, but I'm here all day. So if you have questions, you can also approach me later. So then we'll do questions now, if there are questions.
Thank you. Very interesting talk. I'm a parent of a child with Duchenne's, and I'm interested in what you're explaining between the difference between Duchenne's and Becker's. So what I understand from your explanation, Becker's is where dystrophin is expressed because of some kind of exon skipping and so forth.
No. So I was assuming that there would have been a talk yesterday about this. But I know also that there's many parallel talks, so maybe there was one, but you happened not to be in the room. So what happens is that it's not with exon skipping, it has to do with the mutation. So if the mutation makes the code unreadable, you get Duchenne, because unreadable, there can't be protein produced. Right. But if the mutation keeps the code readable, then you get Becker, because then a protein can be produced because the code is still readable. The only thing that there is a mutation, so the part that is missing, that won't be in the dystrophin protein. So the dystrophin protein will be shorter, but because what is missing is in this middle domain, this elastic domain, if that is slightly shorter, well, it's a problem because you get Becker. But compared to Duchenne, it's much less of a problem.
Right, okay. Yeah. Thank you. It's very interesting.
And I saw many people take pictures, which is fine, but if you want the slides, feel free to email Action Duchenne or me, because I'm happy to share the slides as well, if that's helpful.
Thank you, Annemieke. Thank you very much. There'll be opportunity to ask questions at the end, depending on time, but thank you very much. And Linda, over to you. Thank you.
So, hello. I spoke yesterday, so I might have met most of you yesterday, but I'm here again, so sorry to bore you, perhaps. But anyway, Annemieka's done a brilliant job of going through all the theory and some of the challenges, so hopefully there won't be too much duplication between what I'm going to say and what she said. I've gone for a slightly different approach, so again, forgive me if it's repeating a bit of what Annemieka's said. So I'm going to look at the future perspectives, and I'm going to approach it in a similar way that I did to the exon skipping yesterday talk, if you were in that, so looking at what the challenges are and what's being done to try and address them. So I won't talk about exon skipping, Annemieka's done that already, and I did it yesterday, but for the gene addition, I see the challenges, as Annemieka's pointed out, the immunity, the safety, the functionality of the protein that's being expressed, and also the cost. And then for the gene editing, I don't know why they've put this here. It covers half the slides, doesn't it? Anyway, for the gene editing, it's the applicability, so it's a mutation-specific therapy. And again, it's the safety, efficacy, and delivery issues, because we're using AAVs, as Annemieka's gone through already. So I'll go through my understanding of what's being done to try and address the issues for the gene additions. That's the AAV microdystrophin. So there's a lot on this table, I do apologise, but I will go through all of it in turn. The only thing I won't talk about is what's being done to address the cost of the therapy. People are working on optimising the production methods so it's more efficient and more vector can be produced, but something that will come up in the other issues is treating patients earlier, so that a lower dose can be used. As Annemieke says, it's per kilogram of body weight, so if you can treat earlier, you don't need as much vector. So that is another way of trying to address the cost of the therapy. So if we look at, so I'm going to combine the strategies for addressing the immunology and the toxicity issues together, and there are 4 things I think that are being done. So immune suppression, so as Annemieke has laid out, that is the real major risk factor with AAVs because of the strength of the immune response, not just to the AAV, but also to the microdystrophin. So we can suppress that response, we can tolerize, and I'll go through that. We can use lower doses, and we can use different serotypes, so that's the number that's given to the AAV, so the 9, 8, etc. So we can generate new serotypes. So this is a complicated diagram, and I apologise for that, but it just shows you how complicated the immune response is. So again, as Annemieke has laid out, the AAV is a virus. And, most of us are infected with the virus. It causes like the common cold, different forms, and we will have raised antibodies to those viral infections. So, 30 to 60% of us already have antibodies, including patients who might be treated, to the AAVs that are going to be used. And they will obviously mount a really strong immune response to the AAV and clear it. So that's why patients already have antibodies are precluded from trials. So that cuts down the applicability. But, and again, as Annemiek's laid out, there is going to be an innate immune response, which is on the left-hand side. Because you're putting something that's not normally put into the body will generate a response to try and clear it because it sees it as foreign. So you get something called complement activated, which is seen actually in clinical trials and causes the major issues that are being seen. And this then triggers an acute inflammatory response and then a chronic inflammatory response. So you get clearance of the AAV and then the microdystrophin. So these are real issues that need contending with. And as Annemiek's laid out, from the trials, we are learning on what needs to be done to make AAV a safer option for patients. So Sarepta is just an example. Sarepta, modulated their protocol on what they saw in the early treated patients. So they now include an immune suppression. So they modulate the immune response by suppressing it with drugs. So they give prednisolone, which is an inhibitor, at the beginning of the treatment and taper it off gradually to make sure that the immune response is gone. So that's inhibiting the left-hand side. For those people who have antibodies already, they are, like I said, are precluded from the trial. But obviously, we need to make this a therapy that could be used in all patients. So many researchers are working on trying to think of a strategy to allow them to be included. So you can do a process called plasmapheresis, which is basically the blood is filtered and the antibodies are removed before AAV is administered and put back in, obviously. You can also use what's called empty AAVs. So basically, they've got the protein around the outside of the vector, which normally would trigger the response. And if you give loads of that, or the protein itself, you will mop up the antibodies. So hopefully your AAV with the microdystrophin isn't being cleared. So another thing we could do is tolerization. So again, I apologize for the complicated diagram there, but so it's T cells are the part of the immune system that mount the antibody-directed response if you've been exposed to the antigen before, the trigger before. So we can switch off these T cells with various strategies. And the one that people are examining is using what's called the regulatory T cells on the bottom. So these act to block the activated T cells so that they don't mount the immune response. So this paper was from a few years ago, 2015. So what they did was feed before delivering the AAV. So they would feed the subject with the gene that they're trying to deliver, so the protein, so that they recognize it as self. And you can also feed with the AAV capsid, the bit around the outside. So this is called oral tolerization. So we all can eat all foods generally without any issue, and that's because we've gone through this process of oral tolerization with everything we eat. Obviously, there are some people who are still sensitive to maybe lactose or peanuts, etc., but normally you get tolerized to what you're eating. So, this group examined doing this, and I think this could hold real potential for AAV tolerization and microdystrophin tolerization. So it works, so the protein is obviously taken up through your gut and exposed and taken up by these what are called dendritic cells, which processes them, and they are responsible for making these T regulatory cells, which then act to block the T cells, which would normally recognize the AAV or the microdystrophin, and so you get tolerization. So this could come through in time, for the immune issues that are being seen in trial, and also to allow those patients who already have antibodies to be treatable. So, as Anamika laid out, we're going in with really, really high doses into patients because we need to get to all the muscles in the body, and there's a lot of muscle to treat. So the doses generally used are between 1 and 3 times 10 to the 14 vector genomes per kilogram. And I don't know what that number is, but Anamika called it gazillions or something, so whatever those number of zeros are. So it's a lot of virus going into these patients. So we want to try and go in with a lower dose, and we could do that by trying to enhance the expression of the microdystrophin so that we get more microdystrophin expressed with a lower dose. So we can do that through three methods actually, I think. So all microdystrophins in trial have been through a process called sequence optimization. So the sequence isn't exactly the same as what is normally expressed. It's slightly altered. The same protein, the microdystrophin protein, is expressed, but the sequence is slightly changed, and that's so that it can use some more abundant pieces in the cytoplasm for the expression of the protein. They're called transfer RNases, and some of them aren't there in high abundance, so it limits the expression. But if we can use a more abundant transfer RNA, we will get higher levels of protein expressed. So this has been done already, like I say, for all the microdystrophins, but algorithms changed. It's all based on published data, so it's possible that we could improve the sequence further. And we've done that. We've shown that we can improve the expression levels, but we could also use more specific AAV serotypes. So this is the 8, 9, etc. But we could try and make them more muscle specific by using a different serotype, maybe generate new ones. And we can also use more active and more specific promoters. So they're the bits that drive the expression. So we've been working with the company. They were called Sympromics. They then became Mars Bio, and I think they're now Bayer. They just kept getting bought up. But basically, they use bioinformatics to design synthetic promoters. Again, it's based on published data and machine learning. And we've been testing them to see if we can see an improvement in the expression in muscle. And again, behind the screen, you can see that we do get a really increased expression compared to the control. So there are ways to improve the expression using promoters, but we can also do more homing to the muscle by using different AAV serotypes. So most serotypes have been identified, and the tropisms, where they target is known. But we could generate new serotypes by shuffling up the proteins from all of them. So this is what's shown here on the right to make them look different. So again, they won't be recognized as in the immune response. So it will make them safer, and we can hopefully go in with a lower dose. So just to point out, that's the AAV on the left there, and the capsids are the bits that stick out, so that's what is recognized by the antibodies. But if we can make them look different, they won't be cleared. And again, if we can make the protein more muscle targeting, we can go in with a lower dose. So we're delivering a microdystrophin, and Anamika went through it very well, but I'll do my take on it. So we are delivering this microdystrophin, which isn't fully functional. So at the top there is the full-length protein, which has the two, and I really liked Anamika's analogy to the climbing clips. So we've got one clip and the other clip, so it's the actin binding domain on the left, which is important, and then the orange is the dystroglycan binding domain on the right of the protein. So the three microdystrophins in trial are shown here. So they have the green and they have the orange, the important clips at the end, and then different parts of the middle section, which, has the important parts, but there are bits missing. So if we look at how these would be in the muscle, so on the left there at the top is the full-length dystrophin shown in grey, so you can see the ends, and then the repeat segments in the middle. So they're interacting with a lot of proteins in the muscle. Now, for the microdystrophins, the different variations are shown here on the right. You can see that there's a breakdown in the connections. You know, the actin, the red, isn't binding to the middle of the dystrophin anymore. And in some of them, there's not the green circle, and within the membrane itself, which is the sort of line across the middle, there are connections missing. So it's not fully functional, and that's important. We're only delivering a Becker-like protein. So we could try and improve that with various strategies, and this has been done. This was done a long time ago. But rather than using a single AAV, we could use 2 or 3 to deliver more of the coding sequence. So Lee et al. Used bigger sections in 2 AAV vectors. It's still not the complete protein being delivered, but they used what's called protein transplicing. So that red and the blue line come together and splice the 2 parts together, and we get a longer Becker-like protein expressed, and it worked very well in the MDX mouse. So we've also worked, and others have, on using 3 AAV vectors. So we've got the full DMD gene, and we've divided it into 3, and we're using 3 AAV vectors to deliver the full sequence. And we've got splicing signals, the joining signals, to glue them together to give the full-length dystrophin. So we published the initial study a long time ago, but it was very, very inefficient. So with some Action Duchenne funding, we looked to improve the joining together. And you can see in the sections through the muscle on the bottom there that we are getting some dystrophin-positive fibers, and they do seem to be protecting the muscle from weakness during repeat contractions, and we're increasing the strength. So it works, but it could be improved dramatically. And when you think about it, you know, going in with a single AAV into patients is causing some serious toxic and immunological side effects. So going in with 3 is going to be very, very unlikely. So we haven't been able to get more funding to develop this because of that issue. But as Annemiek's pointed out, we could also use other delivery vectors. So we're working with a company called Replay, who have this herpes simplex virus, so that causes herpes normally, but obviously you gut it and then package in the full-length dystrophin gene and hopefully deliver to muscle. So we'll see where we go. It's just started that project. So you could use other viral vectors that have a bigger packaging capacity. And as Annemiek pointed out, there are nanoparticles which may or may not get to muscle very well. There are different forms shown here. The nanoparticles are in the top left there. So they basically encapsulate the DNA that you're trying to deliver, and you can decorate it with what are called ligands. So basically they're just small peptides, a bit like the cell-penetrating peptides that are being used for the antisense, to give targeted delivery to the muscle. So we're working again with another company called 4Base Bio, who have a patented nanoparticle. They call it a non-viral vector rather than nanoparticle, but it's called Hermes, so hopefully it will deliver nicely to muscle. And they also have a patented DNA synthesis, which is linear DNA rather than circular DNA, which is normally used for generating the DNA copies. So lastly, people are looking to use what are called exosomes. So these are small vesicles, so they're released normally from cells, and they carry sort of messengers to neighboring cells to deliver the message. And these can be harvested, spun down, and then you can load the DNA that you want to deliver into those and give to the patient. So Evox were interested in doing that for Duchenne. I'm not sure where that is now, but people are looking at exosomes. So I'll move on to genome editing, and there is some overlap, obviously, with the AAV side of things, because they are being used to deliver the CRISPR-Cas. But these are the issues: the applicability, the efficacy, safety, and delivery. So applicability, it's a mutation-specific, and it's very inefficient, as Annemiek's gone through. And the risk with CRISPR-Cas is that it's not just going to cut where you want it, it can cut elsewhere, so we need to improve that. So to improve the applicability, so Annemiek told you about cutting out a single exon, but we could target around a mutation hotspot, and people have done this. So Charles Gabash, so he's targeting, either side of a mutation hotspot. So he's cutting out exons 45 to 55, and that would be applicable to 62% of patients. So he's published on that, but it was only done in cells. It didn't move any further forward, and that's because the efficiency was very low. You have to obviously have the cuts on the same piece of DNA, and the efficiency was low, and then you need the right repair to delete that middle section. So we could also do what's called integration of super exons. So this would be to try and repair the coding sequence. So he was targeting Charles Gabash again, I have to say. So he was targeting on the right of exon 51 because it was for an exon 52 deletion. And he was integrating exon 52 to repair it, but he also used what's called a super exon. So it's going from exon 52 right to the end of the gene, so to exon 79. And he was able to show in a mouse model, the deleted exon 52 mouse, that he could get this integration, but it was really, really low efficiency. So that needs improving. So that would be applicable to more patients than the mutation hotspot. But for a therapy or a gene editing therapy that would be applicable to all patients, we could try and integrate a microdystrophin or a full-length dystrophin into what's called a safe harbor site. So there are positions in the human genome where you can cut and integrate without any side effects. So they are there, but you need to know where they are, and you obviously need to target them specifically. So we've been targeting, and again, this was a very short project funded by Action Duchenne. We were targeting what's called the AAVS1 site, and we were delivering this repair template that had the microdystrophin cDNA in it and a promoter. And we were able to show on the RNA level at the top there that we're getting integration, and we are getting some level of protein expressed. It was very weak. So again, it was really inefficient, and we only did this in cells. So again, it needs optimizing. And it's the efficacy of gene editing that needs to be improved. So I won't go through delivery using AAV vectors or using nanoparticles, because we've gone through that with the gene addition. But I'll try and talk through enhancing the repair efficiency. So when we're trying to knock in something, as Anamika said, the muscle cells aren't dividing, so trying to get what's called HDR integration is not going to happen in muscle. So we need to try and exploit the glue that Anamika described it as, the glue sticking the ends together. So we can use various strategies shown there on the left. I won't go through them all, but basically gene editing using CRISPR is a really rapidly moving field. Every week virtually there's a new tool being described. So things are improving, but it's all on the bench science level rather than clinically. But we can exploit that to try and develop a more efficient repair strategy. So CASs do have real serious safety concerns, and we can address that with all of these shown above, which I'll go through very quickly. So the first is using more stringent CASs. So the widely used CAS is called SP-CAS. So for a CAS to cut, they have to recognize a sequence within the genome. They bind, and then they need that sequence beside where they're binding to do the cutting. So for SP-CAS, the widely used one, it's a really simple sequence. It's just NGG, so it's 3 nucleotides on your DNA. And that NGG occurs once in every 32 base pairs, so you're quite likely to get non-specific cutting. So we could use these other CASs, so they're all bacterial proteins, and different bacteria express different CASs. And you can see that they're what's called the PAM sequence, is the bit that they recognize, is more complicated in some of them, which is good. So we'll cut down the off-targets. But it does mean that designing where we want to cut is more complicated, so you need to get a balance. But, we could use a more specific CAS. So this was mentioned, well, not mentioned, but it was on the slide for improving the efficiency. We could also use what's called nicases to make them safer. So Cases, are scissors. They cut through both strands of the DNA. So DNA has got two strands of sequence that are wound around each other. So we can use what are called nicases, which will only cut one strand. And if we pair them, we will get, and in the right positions, we could get cuts that are near each other on opposite strands. And when they are near each other, they will then be repaired, and we can exploit the type of repair because of the sticky ends to make it more efficient. But it means that, it's unlikely that you will get one nicase pairing with the nicase anywhere else apart from where you're trying to target, because they're two different sequences. So we can use that, and it does improve the safety dramatically, preclinically. Or we can also use a completely inactive Cas, so they won't cut any of the strands, but they will bind. And again, we use them as pairs, and we can link the Cas to another cutting enzyme, and it improves or rather reduces the off-target cleavage 10,000-fold. So there are things being done to improve the safety of gene editing in terms of off-targeting. So as for AAVs, they are, Cases are related to bacteria, and we all get infected with bacteria, so many people have antibodies to the Cases that will be used. So we could use similar steps that are being used for AAV, but we can also engineer the Cas to make it look different, like we're doing for the AAV serotypes. That is being looked at as well. And because trials are now happening for liver, we know we can get an understanding of what the immune response is going to be like. So lastly, controlled Cas expression, so Annemiek talked about this, but we can also, Cas, if we get a transient expression, so it's just there for a little while and then disappears, that is much safer than having permanent expression using an AAV. So using the nanoparticles would be great, but we could also deliver the protein in these lipid carriers so that they get turned over. But a way of restricting the expression is to use what are called inducible promoters. So you can get promoters that are switched on by light or switched on by using an antibiotic, so we can just give transient expression and then it disappears. So this is all being looked at and developed. So this is the future perspectives as far as I'm concerned. So I think AAV microdystrophins are really exciting. We are seeing high levels of expression in clinical trials, and we know that Sarepta are filing for approval. So it does work very well, but there are serious safety concerns which need to be addressed, and we are learning from the trials as they happen what needs to be done. We're only delivering the microdystrophin, so we do need to look at other delivery options to be able to deliver more functional dystrophin, and that is being done. For the genome editing, as Annemiek has laid out, that is a really good research tool. The chances of it getting into clinic is very slim because of the safety issues, but, like Annemiek said, there are many people working on it, trying to make things better and safer. And I will stop. Thank you.
Thank you. I think we've got some time for some questions, if anyone has any for Annie, Mikey, or Linda. That's okay. There's one here. Thank you.
It's more of a general question about the slides. Will the slides be available at the end of the conference? Does anyone know?
Oh, yeah. Yeah, I don't know whether... Oh, hello.
Hello.
We're being recorded so that absolutely all the sessions will be available, online after.
Fantastic.
Yeah.
Don't mind me sing.
Thank you. Does anyone else have... Oh, hello.
Hi, I know you mentioned earlier when we were talking about gene addition and gene therapy that boys with early deletions are excluded at the moment. Is that something that might change in the future, or is it that it's not actually possible for the boys with early and large deletions?
I think so. It depends on the type of approach. So we used to say gene therapy, it doesn't matter. So the gene addition, the microdystrophin, we used to say it doesn't matter because it will work for all mutations. Well, it does, but we know now that if you miss the beginning of the gene, then you get this immune response. So that means now that it's no longer applicable for all mutations, but for all except that subgroup. For exon skipping, if the mutations are in that first clicker or in that last clicker, then we know that even if the code is readable, you will get Duchenne, you won't get Becker, because you need these clicker domains. These mutations, fortunately, are relatively rare, but still they are there, and for these patients, exon skipping wouldn't work. And then for the genome editing that people are now mostly working on, they're really trying to do exon skipping with genome editing, so exon deleting. There the same would apply. But these newer methods that Linda also presented on, when you try to replace things, then of course that would be possible also for the very large deletions, or when the deletions would involve these clicker domains. And I'm not sure if Linda has, no?
Yeah?
Hi. One of the questions that I have is, in terms of the mechanism of the dystrophin production, do we know which exons are responsible for the different parts in the pathway? Because it's just that because my son has got out of frame 3 to 7, and he's 14 and he's still walking and he still plays sports and everything. So his consultant at the moment thinks it could be an intermediate type of Duchenne's or a Becker's type Duchenne's. Duchenne's, sorry, excuse me, either an intermediate Duchenne's or a Becker's. And am I right in thinking that if you have Becker's, it means that you are still producing a certain amount of dystrophin, but not as much as, say, like a normal person would?
So, well, there's several things. So first of all, Becker patients make dystrophin, but they make it in anywhere from 13 to 100% of levels. So some make shorter dystrophin at 100%, some make shorter dystrophin at 13%. And we used to think, well, if you make 100%, you should do better than if you make 20%. That's not the case. So having a certain amount apparently already is enough to have Becker. So that's the first thing. Then the second thing is that this deletion, the 3 to 7 deletion, it's a very strange one because it's been found in Duchenne, it's been found in Becker, and it's found in this intermediate. And what happens is, so we know exactly which part of the which exons code for which part of the protein. And what happens in the 3 to 7 is different. It's not exon skipping. You have the protein or the code, normally exon 1 to exon 79. And exon 1, there's a signal that says start the translation here, and then 79, protein complete, stop. Now, there are other starts in exon 8. And what's most likely happening in some patients who have a 3 to 7 deletion is that a start in exon 8 is used. So then the first part is missing from 1 to 8, but 8 to 79 is there. And then the question is, why do some people do this and others not? Because if we knew that, then of course we could make the ones that are not doing it do it. That we don't know, but if your son has, say, a more intermediate, then most likely this is what's happening. And he's making this dystrophin, and it's shorter, and it's, I mean, part of that first clicker domain is, I mean, it's maybe a weaker clicker domain, but apparently enough to have a slower disease progression. So this is common, and this is how that happens for mutations at the very beginning of the gene.
Yeah. Okay. Thank you very much. That's very useful. Thank you.
Does anyone else have a question?
And I think I gave a presentation about the different types of mutations and what that taught us about dystrophin production at another Action Duchenne meeting. I think it was online, so it might still be available. I see people nodding.
Yeah.
So have a look if you're interested to learn more.
Yeah. I've got a few questions actually, so from the community and submitted online as well. I know I've got one, actually both of you and Linda as well. You mentioned about, so what is the alternative for boys who have autoantibodies to AAV? What's the next step? And I know you talked about plasmapheresis and empty AAV. What is the status of that? Is that already in use, or is that the future?
So it's not in use for AAV microdystrophin. I think it's possibly for some liver AAV gene addition therapies, but it's definitely being developed. So it's at the research stage. So, you know, in time it might come through. But yeah, I think people are looking to try and address the immune reaction.
Right. And there's another question which possibly isn't strictly gene therapy related, but it's a general question. But I've received this quite a few times now. It's about the location of the mutations or different exons involved and whether they drive disease severity. Are some mutations more dangerous than others?
Yeah. Again, I'm going to defer to that presentation that I gave. I forgot whether it was last year or the year before. And if people have, I'm happy to answer questions if people email me. I'm stalked enough online to know that my email address should be findable. But yes, definitely there are mutations that we know are less severe. So the mutations at the beginning of the gene that the mother just asked about, those can be less severe. Not always, but they can be less severe. Mutations that need exon 44 skipping to make the code readable are less severe, and that is because all of us skip exon 44 in about 1% of our dystrophin transcript. Doesn't matter, means we make 99% of dystrophin rather than 100. Doesn't matter. But for people that need exon 44 skipping, that means that they now make a little bit of dystrophin, while other they would not. So they have a slower disease progression as well. And then for the rest, for most of the rest, it doesn't matter. So you would think having a mutation at the beginning of the gene is worse than having it at the end. As long as that second clicker domain is not there, it doesn't matter whether the mutation is at the beginning or in the middle, doesn't matter. It does matter for brain involvement, because there, the further to the end of the gene the mutations are, the more likely it is that patients also will have more severe brain problems. And that is because there's also dystrophins in our brain. There's the long dystrophin that's in muscle. None of the Duchenne patients has that, and that's why often there's learning difficulties and other things. But there's also shorter dystrophins that some patients can make and others cannot. And those that cannot make it, they generally have more severe learning difficulties and behavioral difficulties, etc. So for the brain, it matters more than for the muscle in that regard.
Thank you. I just have one last general question. This was asked as well. To both of you, as a clinical researcher for DMD, if you could have one wish that would make your job easier, what would it be?
Wow. Well, I'm not a clinical researcher. I'm a mouse doctor.
Yeah.
Never had any complaints. They're all doing quite well, but they're not that sick to begin with, so that makes it much easier. I think so. I've been in this field for 22 years now. And I think, so looking back, a lot has been achieved, because when I started, we could never envision that AAV gene therapy trials would happen and that they would treat the whole body. We were working on trying to treat individual muscles or limbs, which was a huge problem. So a lot has been achieved, and we didn't even consider that things like genome editing were there. So a lot has been achieved, but on the other hand, the institute I'm working for, we were one of the pioneers of exon skipping. We have patents on exon skipping. It's approved in the US, but it's approved on really minor levels of dystrophin, and I don't know whether that's enough to really make an impact. So I think my wish is that there is something that actually does make an impact on Duchenne patients. And well, I would love that they're cured. I think with the current technology that is not achievable, but anything that can make an impact can slow down the loss of the next function. I think that's my more realistic wish for the near future.
Well, money helps for doing research, so more money and more people who want to be doing research would be amazing. But I think, as Annemieke said, we have made major steps forward as a community of scientists, and it's through support of the Action Duchenne and other charities that we've got to this stage. And I think there is hope on the horizon. We are seeing approvals and, really important clinical trials. So, we have come a long way and the future is still bright. So, it's getting there and we're all working together, which is the important bit. There's a big community.
Thank you very much. Do we have any other questions now? In that case, thank you very much for an engaging talk.